Chromosome 1 is one of those really central genetic features of multiple myeloma and other cancers, by the way, but really in myeloma, 40% of people have it. And for the last decade, people have been looking for the gene on chromosome 1 that causes it to cause adverse outcomes. Unsuccessfully, I should say, but there are maybe 10, 15 papers that have claimed to have identified different genes along chromosome 1. So the idea of this study was to say, is there a single gene on chromosome 1? If not, are there multiple genes? And how does the chromatin structure of chromatin 1 affect translocations and gene expression? So we've done a big study, which I think is really interesting, that shows when you map the sites of copy number change, translocations, copy number loss, you find different regions. It's not just a single region. There may be 7 different deleted regions and at least 4 gained regions on one Q. So now we're starting to split up chromosome 1 into deletions on the short arm, where there are 3, and then regions of gain on the long arm, of which there are 4 plus. So then the question comes is, how were those genes deregulated? And so we mapped them to chromatin structure and topologically associated domains. And you see that the ones that are deleted are in kind of non-expressed regions that are kind of inactive, whereas the ones that are gained are in regions where there's lots of interactions and these genes are overexpressed. And so if you put that data with the gene expression data, pretty much it tells you that there is no single gene on chromosome 1 that causes the problem, that there are multiple genes associated with these different breakpoints that have different impacts. So the reason it's so powerful as a prognostic factor is because it deregulates a whole series of genes that do a lot to the cell biology to make it more aggressive. And if we can find out what these things are doing, then there's the potential to treat it. So I think it's very interesting, that changes the paradigm about the role of chromosome 1 as a prognostic factor.